Good morning. I welcome you all to this second session. So we have three speakers. First speaker is Dr. Rhoda Hawkins.
Thank you. So before I start, I'll just let you know where I am. So I'm still affiliated to the University of Sheffield, but I'm currently at AIMS Ghana, African Institute for Mathematical Sciences in Ghana. So I'd like to start by saying what an honor and privilege it is for me to have been invited to speak at this conference in Sriram's honor on his birthday. So I'm sure I speak for many of us here that Sriram has been a very significant inspiration and influence on my career, particularly his science, but not just his science, also who he is as a person. So Sriram, thank you, and happy birthday. So as my title said, I'm going to talk about active actin networks. So to acknowledge the people who've worked with me on this, I have the photographs of various current and former group members of mine who have contributed to things that I will talk about, and also some experimental collaborators. So we've seen this video before. It's my favorite video of all time. This is a white blood cell chasing a bacterium. But the reason I'm re-showing this video is that the bit I'm interested in is how does this white blood cell eat the bacteria? That process is called phagocytosis, and I'm going to be talking a bit about phagocytosis in this talk. So this video has inspired much of what I'm interested in my career, both in terms of cell migration, how the cells move, but also more recently, how the cell eats bugs. So in order to look at the physics of what's happening in complex processes like this, we need to look at the material properties of the cell itself, and particularly the cytoskeleton. So I'm going to be focusing today on actin, which is one of the components of the cell cytoskeleton. I'm going to talk a bit about actin-myosin contractility, and I will talk more about actin polymerization, and I will apply these to the role of actin in phagocytosis as we think it might be happening. And then I will end with some thoughts on potential future modeling work at a continuum level. And I'm privileged to talk in front of an audience who may well be able to give me plenty of suggestions on that final point. So this is an introductory slide about actin. So what's labeled here in green is the actin cortex around the edge of a cell. And then this is an electron microscopy picture showing in more detail the very dense and branched network that actin forms at here in the edge of the cell, in the cortex. So if we look in more detail at actin itself, it's a filamentous protein. So this is a single actin subunit, and it polymerizes into filaments. And so this is a short section of an actin filament to show you what it looks like in terms of the X-ray crystallography. So these little filaments grow into big networks, and it's at the network level that they are active. So they're active in two different ways. So there is active polymerization. That is due to the fact that they consume ATP to polymerize and grow longer. And then the other aspect in which they are active is in the way that they interact with molecular motors. Actin interacts with molecular motors called myosin. Here is a cartoon showing the interaction. We've got a cluster of myosin motors depicted in yellow here, and they are interacting with two actin filaments. Actin filaments are polar in the sense that the motors will move along the filament in a particular direction. So the filaments, if we go back to what they look like here, they have what we call a pointed end and a barbed end in terms of their shape. And this dictates much of the asymmetry of their processes. So polymerization happens preferentially at one end and depolymerization preferentially at the other end, although depending on the concentrations of monomers, you can get both processes at both ends. And then in terms of the molecular motors, they will move preferentially along one particular direction of the actin filaments. So when you have a situation like I've drawn here, where you have two filaments pointing in opposite directions and a cluster of molecular motors interacting with them, then you get contractile stresses. And that has been very nicely modeled using active matter contractility hydrodynamics. So that's depicted here. So we can describe the system of actin and myosin as a continuum, single component material. I'm calling it actomyosin. That's a common word within the biology field as well. And here I've modeled it as an active polar fluid. And as you will see from my open questions at the end, it's not totally clear to me whether this should be polar or pneumatic. But the reason that I'm describing it as polar here is that I'm going to be putting boundaries into the system and worrying about the anchoring at boundaries for which polarity does matter. But if you're in a bulk system, it's not clear to what extent actin networks are polar or pneumatic. And as with many things within biology, there are probably situations in which they are one or the other. So these are this is the model that you know well, so I'm not going to dwell on that. But what I'm going to show you briefly is some things that my group have done with this. So first of all here, this is slightly off topic, but I wanted to mention it. And it fits quite nicely with the previous talk we had on inclusions, except that it's quite a different kind of system. Here I have a finite system with boundaries, and I have a single inclusion. So this circle here is a passive inclusion, and then I have a thin layer of active polar fluid surrounding that. And the thickness is an output of the calculations and not an input. Now, the motivation for this is that this is a simple model of a cell with a nucleus inside it. And the reason that we looked at this is that I was interested in how does the actomyosin material propel the cell and the nucleus inside it. And what I wanted to highlight here is that our results depend crucially on the polarization anchoring that we put in. So we put anchoring of the polarization at both our boundaries. We have an inner boundary, which is the boundary with the model nucleus, which I should hasten to say is just a solid sphere that's rigid. There's no model for the nucleus in this. And the other boundary at the edge of the so-called cell. So in this top picture, in the large one, we have the polarization depicted here. And in the bottom ones, we have it depicted here. So it's perpendicular and parallel, the two different boundaries, whereas here is perpendicular outwards and inwards. And what I want to highlight here is that the position of the inclusion with respect to the fluid around it is different in these different cases. So in this case, it's more to the back. The arrow here is the direction of movement of the entire system. And in these two versions, it's more at the front. So I just wanted to mention that because I'm going to refer back to it later in my story of phagocytosis. But that's something that we have done using the active polar fluid model, but putting it into a confined system with specific boundary conditions. And all I really want to emphasize at this point is that the boundaries matter a lot, and it changes the behavior of the system. Okay, so now I'd like to switch gear and think more about the polymerization of actin filaments. So actin filaments can polymerize by subunits adding on. They can depolymerize by subunits coming off. And as I said before, one end, which we call the plus end for convenience, grows more, and the other end shrinks more. So you generally get polymerization at one end and depolymerization at the other. But the exact rates of these depend on the concentration of actin monomers that you've got in your system, which can be manipulated if you're in vitro, and it varies a lot within the cell. So if you balance the rates exactly, then you can get what is called actin treadmilling, where the length of the filament stays the same because the rate at which subunits are added at one end equals the rate at which they fall off the other end. So you get a constant length, but you get this continual turnover of monomers within the filament. What I'm going to be talking about mostly is the state in which the filaments are growing, polymerizing, because that is important in phagocytosis. So that was the simpler picture. This is also simple compared to the pictures that biologists draw, but it shows a little bit more of the complexity of this system. So far I've talked just about actin and about the molecular motor myosin, but there's actually a whole plethora of proteins that interact with actin, affecting its dynamics. So in this cartoon, nicely drawn by my PhD student James, we've depicted various proteins that are important for our modeling. So these do different things. In particular, the pink ones here are called ARP23, and they nucleate branches off an existing filament. So you have an existing filament here in yellow, and then you get a new branch forming. And that turns out to be important in the story that I'm talking about. But you get all of these different proteins that control and change the dynamics of the network. And this can get very overwhelming as physicists, as biologists will talk about many different proteins, and there's different drugs that affect these different proteins. And sometimes not enough is known about the different proteins and the drugs that are affecting them and exactly what they're doing in order for us to model correctly. And sometimes we learn the hard way, as I'm discovering at the moment with a particular collaboration of mine, whereby the experimentalists used a particular drug to affect the filaments, and then we realized that the drug was not doing what we thought it was doing, and actually the drug has multiple effects, and it gets very complicated. So that's just a bit of a warning that these things can get a little bit messy. Okay, so here is a nice simple experiment that collaborators of mine in Sheffield did, whereby this is turf microscopy. Let me go back and rerun it. And these are actin filaments growing. So they're very small actin filaments growing longer. So this is an in vitro experiment. So they've put in actin monomers and ATP, and they're just watching them growing. And cunningly, we've stopped the video before it gets too complex, because what we were doing with this video was actually tracking the length and measuring the speed of polymerization. But if you let it run longer, both with the experiment and also with this simulation, then you get much more complex networks forming. And in particular, we can add ARP23 into the system, which is the branching protein, and then get branched filaments. So this here, let me re-show it to you, is a simulation, a stochastic simulation of this experiment of actin filaments growing. Put into this simulation is Brownian dynamics and a Monte Carlo method. So we use Brownian dynamics for the diffusion, and we have a Monte Carlo method for the polymerization and depolymerization of the filaments. So that's the way that this has been set up. And we can add all kinds of things into this simulation. So we can choose what we have and what we don't have, which is very nice. It's something you can't do so easily inside a real cell. So we allow the filaments to bend, to fluctuate, and to branch. We can also add in other effects as we like. Yes, Christian?
Yeah, so the Monte Carlo, is that a kinetic Monte Carlo or just normal Monte Carlo?
It's a Gillespie algorithm.
Okay.
Yes.
Question: is ARP23 also acting as a crosslinker, or do you have separate crosslinkers here?
So we actually don't have separate crosslinkers here. So the only crosslinking is the branched ARP23. But in a real system, you have multiple different crosslinkers as well. And if we wanted, we could put those in, but we haven't for simplicity here.
Can you get entanglement?
Yes, you can get entanglement. So the other thing to say is this, this simulation is 2D, and that's okay with this basically 2D experiment where the, I should have said, the filaments are tethered to a surface. However, later I'll be talking about phagocytosis, and for that it is a problem that our simulation is only in 2D because phagocytosis is a 3-dimensional process. The reason I'm not talking about dimensions there is that in 2D you get volume exclusion much more than you would in 3D, and therefore you get these entanglement effects and things like that kind of exaggerated in the 2D system compared to 3D. Okay, so this is phagocytosis. So these are various cells and bugs. I think if I restart it, you might see quickly, this one went in. You have to watch carefully to see it go in. It's quite fast. Goes in there. And then this is a still showing that process. So in this experimental image from my collaborator Simon Johnson's lab in Sheffield, what we have here is a bug called Cryptococcus. It is a nasty bug for those people who have impaired immune systems. It's the second biggest killer after TB of HIV patients. In fact, it's an AIDS-defining disease. Once somebody has got this disease, then they have AIDS rather than just being HIV positive. So it's a big problem in some countries, and it's not a problem at all for most of us. In fact, this bug is very common in the soil. We've probably all breathed it in through the air. It's been found in right across the globe, but our immune systems fight it off so quickly and easily, we usually don't realize that we have been exposed to it. But for people with HIV and for some people who have a affected immune system, then it's a big problem. So the first line of defense for this infection is the macrophages, which are immune cells that engulf these bugs. And if they do their job properly, they engulf the bugs, get rid of them, and we're fine. So we're interested in this process in terms of how should it work and what might be going wrong when it goes wrong. The other nice thing about this bug is it's a sphere, which is really nice when you're trying to do modeling because it's nice and simple. What we did was we put the same ingredients as I just showed you before for the actin polymerization into this geometry. So what we have here is we have a circle. This is in 2D, I'm afraid. It's a circle which is allowed to fluctuate with bending fluctuations. So it has a persistence length to model a membrane dynamics. So it's a 1D membrane rather than a 2D membrane, but to the best of our ability, we've put in parameters that would mimic a membrane. And then we put in nucleation points for actin and then the polymerization dynamics using this hybrid Brownian dynamics Monte Carlo method. So what we have here is we have a target in green, which is a circular bead or bug, and we allow nucleation just when the membrane is touching the bead. So that's why you only get that actin polymerization at the point where the bead is. We're ignoring what's happening in the rest of the cell. And so what you see here in more detail is you see these filaments growing, and you see branches, you see them fluctuating, and you see this network building up that is pushing the membrane. So what happens overall is you start with this model membrane, then the polymerization builds up and pushes the model membrane out around the bead. I should say that what we're seeing here, it's in the frame of reference where the bead is stationary. So we're seeing the membrane going around the bead. This gets more complicated and a slower process if you allow the bead to move around as well. But for simplicity here, I'm showing versions where the bead is stationary and we've got getting polymerization around the bead. And when we get close, when the two parts of the membrane get close to each other, we artificially allow them to fuse in the simulation and stop the simulation at that point. So we're not properly modeling the fusion process, which most likely uses other proteins to help it along. So this was a proof of principle just to see whether actin polymerization alone could cause phagocytosis of a bug like this. When I say actin polymerization alone, I've not included any specific adhesion here. There's no specific adhesion between the model membrane and the target, and that's different from some previous models that were adhesion-based in terms of primarily adhesion as a mechanism. Most likely both things are happening, but here I wanted us to focus on the actin dynamics because that's where the forces are coming from to deform the membrane around the bead. There are various open questions about what is happening to the rest of the actin. I mentioned the actin cortex at the beginning. Here we've ignored the actin cortex, which I think is okay for the rest of the cell, but at the point of, when it starts, the fact that I've ignored the pre-existing actin is probably an issue. What I would like to find out is to what extent the polymerizing actin is pushing against cortical actin, or is the cortical actin completely remodeled and turned into this polymerizing actin pushing around the phagocytosis target. That's not clear from the biology at this stage, but it matters a lot in terms of the properties of the membrane, because if your membrane is simply a lipid bilayer, that's a very different object from if it is a lipid bilayer attached to an actin cortex. So that's a bit of an open question at the moment. Oh yes, I forgot about this cartoon. That's simply to show the Brownian ratchet mechanism of how the polymerization is actually pushing against something. So that's thanks to an old paper, I think, by Jean-François. Anyway, okay, so how am I doing with time, sorry? Okay, thank you. So what I'd like to mention now is that what I showed you before considers only actin polymerization. And what happens is that actually at the end, it takes quite a long time to do the final step. And that's not what is seen in experiments. What's seen in experiments is that the final stage is really quite fast. It's faster than the first half. So the first half of the process happens at a certain speed, and then the second half, it's sped up. And so we started thinking about what else might be happening in the final stages. Because here we show that actin polymerization alone in this model is sufficient, but it's not very fast compared to what's seen in experiments. So here we're suggesting that in addition to the actin polymerization that I've shown in the previous slides, we also have actin-myosin contraction happening. And that the actin-myosin contraction can cause an inward force to push the bead into the cell faster than you would get just by the actin polymerization. So an approximated back-of-the-envelope calculation gave us this flow speed being proportional to the R is the size of the bead, being linear here, and linear with the speed. But we haven't done the full calculation yet, including the proper hydrodynamics of an active fluid to consider that. But this brings me to a general point, which is that this is an example of a system within biology of which there are many examples where you have actin-myosin contraction and actin polymerization both happening. So you also have both of those happening in cell migration, in cell division. Many systems have both of these active elements happening at once. So I'd like to finish with a few thoughts on where I'm interested in going in the future. So I started thinking about this, but only very preliminarily. So my question is, how can we relate the kind of microscopic modeling that I described in terms of actin polymerization to hydrodynamic models? So actin polymerization can, in my opinion, give us an extensile active stress if it's growing, pushing the other filaments out. So how do we model that within a hydrodynamic model for actin-myosin contractility? Is that effect nematic or polar? How do we relate it to flocking models? If you have treadmilling actin or even growing actin, you have a polymerization velocity. This is like a self-propelled rod. Again, is this nematic or polar? Probably polar in this case. So how is actin polymerization related to these models? And finally, boundaries matter. So boundaries can be like a source term for fluid flow. You can get pushing against boundaries, you can get retrograde flow, protrusions at the cell boundary. I think in the future, we all need to think about boundaries more because they change things. So that's where I would like to conclude. I've talked to you about a little bit about actin-myosin contractility, a bit more about polymerization and phagocytosis. And in the future, I'm interested, and even later today, I'm interested in conversations with anyone who would like to talk about continuum modeling of these kind of systems. And I'd like to leave you with that slide about Ames, Ghana. If anyone wants to talk to me more about Ames, Ghana, or is interested in visiting, let me know. Thank you.
Yeah. Okay. So going back to the simulation of phagocytosis, so you said that there is no specific or addition of this inclusion on the membrane, and yet the actin cytoskeleton is able to sort of polymerize around it. Can you please explain how exactly does it do it? Because without the addition, where does the force come from?
Sorry, I'm just reopening this because it's crashed. So the force is coming just from the actin polymerization. So you might ask, what is it pushing off?
Right.
And it's pushing off itself. It builds up a network and pushes off itself. So what matters is what will move more easily, the target or the cell with the actin network. So either it will push the actin network backwards, or it will push the target forwards, or the membrane forwards. So it's a balance of all of these different aspects.
Right, but my confusion is that the polymerization doesn't start until the membrane hits the target, right?
We start the simulation with the target touching the membrane.
I see, okay.
Yes, so we don't model the previous bit, which is how the interaction happens in the first place.
I see, so that's your initial condition. Okay.
Our initial condition is it's already touching, absolutely, yeah. So in terms of the process of the two coming together, that's a whole different story that we've not looked at. So I think if I just do slideshow. Okay, sorry, I was trying to find the simulation, but we finished that question now. Okay. Yes.
So, for the late stage, when it goes up like this, for the late stage, to, make the late stage faster, wouldn't just making the OP23 recruitment curvature dependent, for instance, do it?
Curvature dependent.
Yeah.
Yeah, I think that's a very interesting idea, and NearGov is already exploring the curvature effects of that. So yeah.
Jacques and Sriram and Toner did.
Yeah, so I've not included that, but I think that's, I think it's especially relevant when your target is small. So one thing I didn't talk about here is we've done a whole study of how this is dependent on the size of the target. And there's an optimum size of target, both in the simulations and in the experiments. And we've done some analytical calculations as well. I don't have the story wrapped up yet, but there's an interesting optimal size that appears. And I think, and so basically, if the target is too big or too small, it's difficult to phagocytose. And in the case where it's too small, I think curvature-dependent effects are really important.
When you talk about this hydrodynamic model and the question of the polarity, would you think that, or is in your mind the idea that all the aspect of the polarization of actin could be included as a boundary condition? That because at the end there you can break this symmetry between pneumaticity and polarity, and then have this coupling to the.
So I know how to put it in as a boundary condition. What I'm not sure about how to do is to put it in the bulk where it's not by a boundary. But I think if you, in terms of boundaries, it can be a source term, like a source of fluid. But in the boundary, and when I say polar or pneumatic, it depends. So actin filaments are polar, we know that. The question is, when they're organized, are they organized in a polar state or in a pneumatic state? And that will affect the polymerization and the effects there. So that's what I mean by, is it polar or is it pneumatic? The filaments themselves are polar, but is the element of our continuum modeling polar or pneumatic, or should it be polar or pneumatic? That to me is not totally clear.
Okay, let us thank the speaker for the nice talk. So,